class. Okay. So people, can you see my screen?
Yes, Sandeep.
People? Huh. Okay, great. Thank you. So, okay. So yesterday we talked about the process of eutrophication and how a lake goes from very clear water with low nutrients to green blue water with lots of nutrients to finally almost brown water with almost zero nutrients, and the death of all fishes, etc., etc. And as we discussed, that this is not a problem that is there only in small lakes. Even the largest of the lakes on the planet can suffer from this. And finally, as we were discussing, that since every human settlement, as well as almost all plants, animals, everything depends on water. Therefore, the eutrophication of water bodies, particularly large lakes, is a huge issue. And therefore, a lot of effort has been spent in trying to figure out why this happens. We discussed some of those, and more critically, how to reverse the process. So this, although this is very important, this process of reversing the trophic status of lakes is something that is of relatively recent origin, and it mainly starts with this particular lake called Lake Washington in Seattle, US. So in the 1940s, this lake was oligotrophic, and it had a very clear water, etc., etc. And then in 1950s and 60s, as Seattle started increasing in size as a city, the amount of sewage that those guys were generating, that increased. And obviously they had such a large lake, and those days people thought that sewage disposal in lake is fine, the lake will take care of itself. So they started putting all those sewage into the Lake Washington. And as a result, very soon the lake essentially turned into, for people in Delhi, they will be able to understand this. They turned basically into what the Yamuna is in Delhi, which basically means it's turned into a big mass of black, stinking water. And in this particular case, the stink was so heavy that this particular lake was nicknamed Lake Stinko. So that is when people in the nearby universities, they started studying this particular lake, trying to understand that why is it that the lake is becoming like this. And the one person who did more along this line than anybody else was this man called W.T. Edmondson. So he was the one who studied the profile of the lake over time. This was a very long-term study, and ended up figuring out much of what we understand about how human wastes end up speeding up eutrophication. Now, most scientists would have stopped there, but W.K. Edmondson was slightly special because he did not stop with figuring out what was wrong. He actually went ahead and started petitioning. He started, badgering the local representatives, the politicians, etc., etc., saying that you have to do something about it. And because of, well, several factors, but one of the prominent factors in that was all the data, all the insight that Edmondson was producing from his work. And because of all that, people finally figured out that it's not a great idea to pump this sewage into this place, into Lake Washington. And at some point, they started diverting the sewage to the sea. And when that happened, so lots of nice things happen. So this is a slightly complicated graph. I'll walk you through this particular graph. So what this graph is showing is a number of parameters between roughly 1950 to 1985 or 1990 or so, about 40-year worth of data over here. So what happens is that somewhere around the 1950s, the sewage effluent, it starts increasing, and when that increases, then the dissolved phosphorus level. Can you guys see my? Okay, let me see if I can. So can you guys see my pointer? So, so the sewage level went up, and when the sewage level went up, the dissolved phosphorus levels also started going up. Okay, so this is the period during which the transparency level. So transparency is measured in meters. Okay, so what they normally do is, well, there are multiple ways. The most common way of doing it is they take a disk and they divide the disk into 4 equal parts, 90 degree, 90 degree, etc. And then they put diagonals, they make black, and one of the diagonals they make white. So you have 4 quadrants, 2 of them are black, 2 of them are white. And then what they do is they essentially tie this with a rope and they start slowly lowering it into the water. The depth at which they cannot distinguish between the white part of the disk and the black part of the disk, that is the level at which it is said that the transparency has become 0. So how deep do you have to put that disk in order to get 0 transparency? That is what is measured as the level of transparency of the lake. So obviously, the clearer the water, the light will penetrate to a deeper and deeper extent, and therefore you have to lower the disk more and more in order to get that depth. Whereas the more turbulent, the more turbid the water, the disk will essentially become invisible at lower heights. So which is why this thing, if you look at it, it is actually scale is going from top to bottom. Okay. So initially the transparency was somewhere around 2 meters or so, and then as the sewage went up, the transparency actually went down. Okay, that's why these values increased. Then at some point they started their activism, etc. The sewage was diverted, and when this is showing the effluent amount, okay, in percentage. You can see that it didn't happen in one shot. It basically happened in steps. So as more and more lines to the sea were laid, more and more fraction of the sewage started going into the sea. So when that started happening, initially the dissolved phosphorus level for some years stayed constant, and then as the sewage effluent started coming down, the dissolved phosphorus level also started coming down. Okay, when the dissolved phosphorus level started coming down, then initially this transparency became better, but then at some point it kind of hit some kind of equilibrium. Okay, it basically didn't change too much in this zone. Then if you look at it here, sewage effluent has become 0 somewhere around 1968-69 or so. The dissolved phosphorus level, it came back, but obviously it will never go to 0. It kind of stabilized at some level. Now there is no change that is happening in the phosphorus levels over here. Okay, in spite of that, if you look at it, at some point, which is roughly denoted by this dotted line, the transparency levels drastically improved. Okay, and it became very, very transparent up to 6 meters, 7 meters. That's a pretty decent level of transparency. So what exactly happened that in some sense flipped it from this stage to this stage? What happened was, at some point, the crustacean Daphnia came back to the water. Now these crustaceans, they are actually very efficient filter feeders. So these guys, once they appeared, they started eating the phytoplanktons at a very fast rate. As a result, the water became much, much clearer. Okay, so this is what is written over here. And since phosphates had been implicated as being the primary problem source, Therefore, there were a few legislations that were passed around that time, and at that time the primary source of phosphate was detergents that were being used at homes. So the detergent manufacturers, they were forced to change their formulations from going from phosphate rich salts to non-phosphate rich salts. Okay, so this is a very classic case that happened. Now I'm going to talk about a more
Sir,
bolo.
hello.
Bolo.
Sir, I had two questions.
Bolo.
One is we can see that the levels of transparency is going down already before the Daphnia has been included. I mean, in the system. Why is
Yes,
it so?
that is because phosphate has come down. The moment phosphate has come down, now it has become a limiting factor. Once it has become a limiting factor, then the cyanobacteria have started dying. Okay. So the cyanobacteria levels have gone down, so transparency has started improving.
But then when Daphnia comes in, it drastically improves.
Exactly, because see, if you look at this, phosphate level will never go zero, right?
No.
So the phosphate level stabilizes over here. Now this phosphate level is sufficient to maintain this level of. Can you see my cursor?
Yeah. Yes, sir.
So it is definitely it is okay to maintain this level of cyanobacteria population. But then once the Daphnia comes in over here, the Daphnia is what drives the cyanobacterial population further down. And when the Daphnia drives the population further down, the transparency becomes better. Okay?
And also, sir, for in cases of very shallow lakes that are extremely clear, like the ones we sometimes see in Lonely Planet magazines and all. So in those cases, if we even dip that transparency measurement thing, and we are marking it, suppose the depth of the lake is 3 meters, and we are marking it 3 meters clear. But when we are, but then if we compare it to something like maybe Lake Victoria or something that is like very deep, and there you are telling it's deep up to 5 meters. It's clear, transparent up to 5 meters, but we cannot compare those two values, right?
You cannot. That is why in limnology you always deal with shallow lakes separately and you deal with deep lakes separately. Okay. The entire, remember what I talked about yesterday about how long the light penetrates, all the heterogeneity that appears. Okay, so there is also the profile of dissolved oxygen with depth. So that profile is also very different between shallow lakes and deep lakes. So you always consider these two separately. You never mix the two up. Okay.
Okay. Thanks.
Okay. Anybody has anything else? So what I need to do now is to take you guys to a different.
Sir. So how did the Daphnia come back suddenly, so as to speak?
That is hard to say, but see, the point is that this lake is not a closed system, right? It is getting runoffs, it is getting from streams and whatever rainfall, etc., etc. So from somewhere it might have come. It's not entirely clear where it comes from. If you are asking whether it was purposefully reintroduced or not, most probably not. I don't think they did it at that point.
Okay, sir. And
a lot of these things have cysts, right? So when the conditions get favorable enough, or maybe when there's just competition, they just.
That is true, but Daphnia, I don't know whether it has that kind of cyst or not.
And also Daphnia is a pretty like cosmopolitan sort of a species. You can find it quite easily. I mean, we found it in our pond culture slide.
Yeah. That is true. But what he is saying is, if they had all died out, then where did they suddenly come from? Obviously, they did not spontaneously arise. Okay. They probably came from some nearby water body. We don't really know where they came from.
Sir, why did they die out actually?
Daphnia died out because the Daphnia has some conditions within which it can live. So if the water becomes too turbid, then essentially it clogs the filter apparatus of the Daphnia. Okay, and remember, when all these things are happening, the pH etc. of the water is also changing. Remember I said that the pH becomes more positive.
Correct.
I mean, it increases basically. It goes towards the basic side.
So because
So,
of the effluent, the pH increases, so the Daphnia is not able to survive.
pH and more important, and also some of the planktons, they actually end up clogging the filtering apparatus of Daphnia.
Sir,
So
but
until
doesn't
the-
Daphnia feed on the plankton?
they will not feed on every one of them, no?
Okay, okay.
Ha. I mean, everybody has their own food base. Okay, so I wanted to give you guys. Bolo? Sorry, I didn't catch that.
Can you build a model in the lab of these kinds of lakes?
Can you build a model?
In the laboratory?
There's a lot of echo. Can you just write it in the chat box? Yeah, you will. Let me start the video and then I'll look at the chat box. Okay. So everybody can see this video.
Yes, we can.
Beautiful, powerful, and life-sustaining. North America's Great Lakes make up the largest freshwater system on the surface of the Earth. Of the 5 Great Lakes, Lake Erie is the shallowest and holds the least amount of water. Yet it is vital to the region's industries, economy, and public health.
The water itself is used for everything from drinking water to irrigating crops, for boating, for fishing, both commercial fishing and sport fishing. And so really any activity that you think of that uses lake water is affected by the quality of that water.
In the 1960s, Lake Erie's water quality was so bad, some people called the lake dead. Then government policies and industrial cleanup efforts greatly restored its health. But since the 1990s, scientists have once again seen a decline in Lake Erie's water quality.
That's a fish
and
kill.
a resurging problem with algal blooms. In 2011, toxic blue-green algae covered 1/6 of Lake Erie, with the largest toxic bloom ever recorded there, a problem visible from space.
We don't want the Great Lakes to become a cesspool of green scum. We want water that is healthy, enjoyable, and safe.
Tom Bridgman, a freshwater ecologist at the Lake Erie Center in Toledo, Ohio, is participating in a 5-year study funded by the National Science Foundation to evaluate the impacts of climate change, farming practices, and public policy on water quality. Ecologist Anna Mikulak leads the team.
And it's only once we understand how that system is working currently that we can really hypothesize about what types of mechanisms, whether it be governance mechanisms or scientific solutions, might actually help with the water quality of the Great Lakes.
Algal blooms are generally caused by an excess amount of nutrients in the water that feeds algal growth, what's called eutrophication. The most common of these nutrients is phosphorus, found in fertilizers. And how are these algae-feeding nutrients getting into Lake Erie? Scientists point to the combination of 2 primary causes: farming practices, such as spraying fertilizers on the surface without mixing it into the soil, and changes in rainfall patterns. With a gentle rainfall on fertilized fields, the nutrients are allowed to seep into the soil as food for crops. However, precipitation patterns have changed recently with more intense spring rainfall. These heavy rains create more runoff that washes over the soil surface, taking more fertilizer with it into ditches, streams, and rivers that flow into the lake.
And it's only a matter of hours between when water enters a ditch to when it enters Lake Erie. It's a very quick trip from field to lake.
As part of the study, Bridgman and his team are collecting water samples at 6 different locations where water enters Lake Erie's western basin. They filter the water and measure the types and amounts of algae. The water samples are sent to the National Center for Water Quality Research in Tiffin, Ohio, to be analyzed for phosphorus. Water quality specialist Pete Richards, also part of the NSF study, uses the data to understand the complex relationships among precipitation, land use practices, and nutrient runoff to the lake.
To try to deal with only one piece of it can be useful, but it's never going to capture the whole story. So it's something that really has to be done integrating many different disciplines and understandings.
One solution is something called variable rate technology, which some farmers like Brett Margraf in McCutcheonville, Ohio, use to apply different amounts of fertilizers in different parts of the field. Using GPS, Margraf locates specific points in his field where he takes soil samples. The samples are tested for nutrients, allowing Margraf to map the fertility of his field and apply only what is needed, where it's needed.
There's a concept called the 4 R's, which is the right place, the right amount, the right time, and the right method, that sort of encapsulates this idea of trying to be smart about the way we use the nutrients.
To manage the amount of nutrients coming into Lake Erie, scientists, farmers, communities, and policymakers are working together to find solutions for better farming practices and improved water quality on the lake.
The minute that we stop putting fertilizers into the water, it will start to clean itself up. And within a couple years, it would be healthy again. So that's one of the hopeful things about Lake Erie, is that it rebounds very quickly.
Beautiful and irreplaceable, Lake Erie and the entire Great Lakes system are an important natural resource unlike anywhere else on Earth.
Okay. Okay, so I'm not really sure about the last sentence though. Unlike anywhere else in Earth, I'm going to argue that lakes, large lakes are there everywhere on the Earth, and everywhere the problem is similar, etc., etc. But the main thing over here is that this the way, if you use a lot of fertilizer, then the way you put the fertilizer and the way the rain interacts with that fertilizer is very important. So okay, this year most of you are not in Pune, but you guys might have seen that there is a difference between how rain falls in Pune and how rain falls in, let's say, Bengal or Kerala or slightly more coastal parts. So if you see the rainfall pattern in this area, very often the rainfall, it falls for like 20-30 minute duration, and the rainfall is not severe. It's like a very gentle drizzle kind of a thing. And then there are so many other bouts of that throughout the day. Whereas in places like Bengal and Kerala, when the rain falls, it falls. I mean, it really hurts you sometimes if you are standing in that rain. Okay. So obviously, the first kind of rain is when it interacts with the fertilizer, the amount of runoff that it is going to cause is going to be far less compared to the kind of rainfall that you get in those other places. So that's the thing that this guys were talking about. The other important thing that they were talking about is why I chose that video to show you guys, which is that if you take any area, if you take any field, then the distribution of nutrients in it is never homogeneous. You are going to get patches. Okay, and if you are going to get patches, then there is actually little point in applying your fertilizer homogeneously, because then in some areas there is excess fertilizer, and in other areas you may not have reached the amount that you need to reach. Therefore, what these guys were suggesting, and that's what that guy was doing by taking cores from different areas in the field, is that map the nutrient amount in various parts of the field and then apply your fertilizer intelligently. So obviously in this there is a little bit of extra cost, because you have to sample, you have to test it, but the hope is that that cost is made up by the fact that you are overall applying less amount of fertilizer now, and B, hopefully that cost is also going to be made up because you are going to spend less in terms of cleaning up your rivers or lakes later. Now, of course, here you get into a private good versus public good problem, because the fertilizer cost is being borne by the farmer, the cost of testing is being borne by the farmer, whereas the cost of cleaning up the river or cleaning up the lake will be most probably borne up by the local government. So it's not in the economic interest of the farmer to do this kind of a practice, and so on and so forth. Anyway, those are going into a different area altogether. So okay, so now I'll go back to my presentation. Okay. Oh, yeah. Right. So we saw about Lake Erie. Now, in both the cases, you saw that the level of phosphate seems to be the major problem over here. And if that is the major problem, then the solution to that should also be simple. You should somehow reduce the phosphate level. Either divert the sewage, or as they did with the detergents, try to make sure that less amount of phosphate is used in whatever stuff you are doing. Now the problem is, although it seems simple, it's actually in reality not that simple, because as I was telling you guys, that phosphate is also something that the plants need for growth. That is why it is there in the fertilizer in the first place. And while you can replace phosphate in the detergents, you can't really replace phosphate in the fertilizers. It's just not possible. Okay. There is one more issue over here, and that issue will become clear if we look at this figure. So in this, what you have over here on x-axis is the total phosphate level, and on y-axis what you have is the level of, or fraction of, the lake surface which is covered by this kind of a vegetation. Okay, this is what is known as the charophyte vegetation. So typically these are the submerged vegetations that I was talking about yesterday, and typically the more of these you have, the healthier is your pond. That is because these things, they require light for growing, and therefore if they are not there, you can be reasonably sure that there is little light in your pond. So what they did was, so this is about a particular lake called Lake Veluwe, in which they ended up reducing the level of the phosphate. Okay, so as you are going in this direction, the level of the phosphate is going down. So when they are reducing the level of the phosphate, that is what is being represented by the red arrow. Okay, so as you see that when the on this side, right hand side, phosphate level is increasing, you can see that as phosphate levels increased, then the fraction of the surface covered by the vegetation went down. Okay, up to till this point when this almost hit zero. Now after this point, they started reducing the phosphate level. Now very interestingly, when they started reducing the phosphate level, the level of charophyte vegetation did not go up straight away. So if you look at this point, it becomes the clearest. So when they were reduced, when the phosphate levels were increasing, at this value of the phosphate, you still had about 10% charophyte vegetation. But then when you are reducing the phosphate level from high to low, then at this value, you actually still don't have any charophyte vegetation. Okay, and over here, when the phosphate levels have gone to about 0.17 or so, going forward, you had about 20% vegetation. Here the vegetation has barely, I mean, here it is not even there. Here it has barely started coming back. Okay, so turns out that phosphate level alone is not doing this. So if when the phosphate level is increasing, then even high phosphate levels are sustaining some amount of vegetation. But when you are going back, you have to drive the phosphate levels much lower. Okay, you have to, for example, drive it to about 10% or so, 0.10 per milligrams per liter or so, before you start getting some good amount of charophytic vegetation. In other words, this is an example where the state of the system depends upon the path that you take to reach that state. It's not agnostic of the path. So in physics, you might have seen this phenomena or heard of this phenomena by the name of hysteresis, okay, which is what is stated over here. So this is a very nice example of hysteresis in a pond ecosystem. Now the question is, why is it that we get this kind of a hysteresis in this kind of a system? So in order to understand that, we need to understand the various kinds of interactions that are happening in shallow lakes. Okay, so please remember, I told you that in the context of lake ecosystems, you have to understand that deep lakes operate very differently from shallow lakes. So whatever I'm telling you over here is just representative of what is happening in a shallow lake, and whatever I'm telling you over here is the distillate of about 30-40 years worth of work, about, I don't know, a few hundred, if not more than that number of papers about how the different components of the ecosystem interact, all that coming together to form this just one diagram. All right. So it's a slightly complex diagram. I'll walk you through the diagram. So in this diagram, you have various agents, various things that are important for the health of the lake, and these things are connected to each other by arrows, and on these arrows you have signs. Okay, so the signs basically tell you what is it that an increase in that factor is going to do to the other factor. So if it's a negative sign, then they are inversely related, which means an increase in this is going to lead to a reduction in this. And if there's, for example, there's a positive sign, then that means an increase in this will also lead to an increase in this. Okay. So let's start with the vegetation. So this is the submerged vegetation of this type that I was talking about. So in a shallow lake, when the amount of vegetation increases, now what are these vegetation doing? These vegetation, remember, these are rooted vegetation. Okay, so they are submerged vegetation. So they act like baffles. Okay, so because they act like baffles, what will they do to waves? The waves that are being formed, they will reduce them because they are kind of dissipating the force of the waves. That is why if the amount of submerged vegetation is large, then the waves that are being formed in the lakes will go down. Now, when these vegetations, many of them, they end up secreting chemicals, and these chemicals, they very often are something that reduce the growth rate of other species. So this phenomena by which, typically this is used for plants, by which plants by secreting chemicals reduces the growth rate of other species, this phenomena is called allelopathy. And the kind of chemicals that are secreted, they are called allelopathic substances or allelopathic chemicals. So obviously when the amount of vegetation goes up, the amount of allelopathic substance in the pond, in the lake, is going to go up. Now when these allelopathic substances go up, then what will they do to algae? Remember, these are harmful chemicals, so they will have a negative effect on the growth of the algae. Now these vegetations, obviously they also need nutrients, so they are in general going to reduce the amount of nutrients, which is why there is a minus sign over here. Now nutrients by themselves, the more the nutrients are there, the more they will support the growth of the algae, and the more the growth of the algae, these are the blue-green algae which is floating on the top, the more will be the turbidity in the pond. Okay, so this is this loop. Now take this loop. So here, this is the only arrow which doesn't go to a oval, which goes to an arrow. Okay, so there are fishes in the pond. Now these fishes, they end up eating the so-called zooplanktons, which is why they have a negative effect on them. Now, as the vegetation increases, these zooplanktons, what they do is they go and hide over there. Okay, I mean, they hide below the leaves, they hide in the stems, and so on and so forth. Therefore, this negative effect of fish on zooplankton, if you increase the vegetation, this has a negative effect on that negative effect. Okay, so that is why you have this arrow falling on this arrow, and there is a negative sign over here. Now these zooplanktons, they eat the algae, which is why they have a negative sign over here. Now these fishes, remember these are shallow lakes, and if these fishes, they are slightly largish fishes, then they actually end up, and because of swimming in the lower regions, swimming near the surface, and during that process they also end up raking up the mud basically. So if they rake up the mud, then the amount of resuspended sediment in the lake goes up, which is why there's a plus sign over here. And when the resuspended sediment level goes up, that increases the turbidity level in the pond. Okay, and this vegetations, they have a negative effect on waves, and these waves, they tend to, by themselves, if there are more waves, there will be more resuspended sediments, which will also end up increasing the turbidity. Okay, so this in some sense is the total set of interactions. Now, as you can see, this is a network. Okay, this is not a simple linear, A leads to B leads to C kind of a thing. Now, in this network, if you want to figure out what is the effect of a particular component on another component, then all you need to do is to multiply the signs which are connecting those two. So for example, if you want to figure out what is the effect of increasing, let's say, fish on turbidity. So fish to resuspended sediment is a plus, resuspended sediment to a turbidity is a plus. So this is a plus into plus. So increase in fish is going to, in general, increase in turbidity. Okay. Similarly, if you do it now from this side, so if you increase fishes over here, then this is a minus, minus into minus is a plus, plus into plus is a plus. So even through this side, this is going to be a plus. So the way it will work is that fishes will eat the zooplanktons. The reduced number of zooplanktons means the phytoplankton number goes up, and when the phytoplankton number goes up, turbidity obviously goes up. Okay. So is this making sense, this whole diagram, people?
Yes, sir.
Yeah.
Yes, sir.
Okay.
Sir,
Bolo?
I didn't understand how fishes hiding behind vegetation is, reducing the thing of zooplankton.
Fishes don't hide behind vegetation. Zooplanktons do. The zooplanktons, they are very small. That reduces their predation.
Sir, what is the contribution of each of the effects you have mentioned? I mean, how significant is the contribution of the fish swimming to...
What you're really asking is what is the magnitude?
Yeah.
Right. So this, what I'm showing you over here is a qualitative thing. What is the quantitative stuff behind this? Because end of the day, it is the quantitative stuff that will matter. And also, suppose there are two different paths leading to the same thing, and one of them is giving me a plus, the other is giving me a minus, then somewhere I need to put quantities in that to figure out what the net effect will be, right?
Yeah.
That's pretty much the question that you're asking. Yeah, that's a completely valid point. See, the problem here is that putting quantity to this is going to be very, very difficult. Okay, so for example, this bit over here, the fish, zooplankton, algae turbidity, qualitatively it's easy to understand, but the quantity is going to depend on, for example, how much water is there, correct? The quantitative effect is also going to depend on what the other loops are doing and so on. So you can do one set of data, and from one set of data you can create the values, magnitude values for one system. However, coming up with a generic thing for these values is close to impossible, because generically speaking, the values will differ from system to system depending on what the other loops are doing. So here I just wanted to give you guys a qualitative understanding of the whole thing, because that is what is needed to answer this question, why we are getting this hysteresis over here. Okay. But your
Sir,
point is valid.
When we started with fish and we went to resuspended sediments in the turbidity, and then we went through the other side of the loop, right? Like through zooplankton, algae, and then to turbidity, and we showed that both of them are positive. Does this have to be true always? Because when looking at the diagram, from whichever point I take, however I try to go to the other side, whichever loop I take, always is the same positive. But does it always have to be positive from both sides, or can it be like positive, negative, or like?
See, this particular thing that I am showing you is a distillate of several studies on shallow lakes. Okay, so in shallow lakes, in general, this is how it works. But you can always come up with a and I have also told you intuitively why that is so, isn't it? But there are other situations possible where the whole thing might operate differently. Okay, so for example, in deep lakes, some of these things will operate differently.
Okay.
So this is just true for shallow lakes.
For one like thing.
Sorry?
Okay, yeah. So it's possible for one thing to influence, like it's possible for fish to influence turbidity positively from one side and negatively from another side.
Technically, it is possible. But as far as these loops are concerned, here it is not.
Okay, cool.
So we'll just quickly now try to understand with this what happens because of which you get the hysteresis. So what happens is like this. So in the system, in these shallow lake systems, when we have the vegetation, then that vegetation has a very major effect on the turbidity. It suppresses the turbidity, right? However, the vegetation itself depends on the turbidity. So if the turbidity goes beyond a certain point, then the release that you get by not having the vegetation is massive. So look at this blue line. Okay, so as long as there is vegetation in the system, as you increase your nutrients, the turbidity will increase according to this blue line. Fine. However, the moment there is no vegetation, then if you increase the nutrients even slightly, you are going to get a lot of increase in turbidity because of those zooplanktons. So when there is no vegetation, then the relationship between nutrient and turbidity is actually going to follow the red line. All right. Now, there is a critical turbidity beyond which the vegetation cannot exist, simply because there is not enough light and so on. So once this critical turbidity level is crossed, now there is no vegetation. Now this thing is going according to the red line. Now suppose you have started using different measures, and by using those measures you are bringing the nutrient level down. Now when you are bringing the nutrient level down, you are following the red line. You are no longer following the blue line. Okay. So you are bringing it down, you are bringing it down, you are bringing it down. So for example, at these places, the nutrient level is already much lower than what it was before the critical turbidity was reached. However, even then you are much higher in terms of turbidity levels. So you have to keep on doing this till you bring this beyond the critical turbidity level. Only at that point does the vegetation come back, and only when the vegetation comes back do you come back to this blue line, and then you essentially go towards your original state. Okay, so this is what is known in technical language as alternative stable states. So the relationship between turbidity and vegetation, it will either follow this curve or it will follow this curve, depending on whether vegetation itself exists or not. Okay, and this thing over here, where you have a critical turbidity beyond which you change, the relationship changes from the blue line to the red line, the relationship changes from one regime to another regime. This point is what is known as the tipping point. So if you are aware of the work of Malcolm Gladwell, you know that he has written a book on this particular thing. So this is the point at which the phase shift happens. And here what I am showing you, I'm just showing it to you, without any maths. I'm just showing it to you using just 2 lines to explain the concept of alternative stable states. However, you can actually put maths in this, and in this particular paper I'm going to share with you guys, Schaefer et al. Nature 2001. They have given the mathematical model behind this, and very interestingly, they have shown that even if you put in a lot of realism in this, even if you put in a lot of other conditions, this alternate stable state wala part always remains. And therefore, their argument is that because of this relationship, you get this kind of a hysteresis state in this particular shallow lake, Lake Bellamy. Okay? So I think we'll look at alternative stable states in a little bit of detail in the next class. So I think we have run out of time. So what I'll do is I will stop presenting. Yeah, I will also stop recording. Vikram, this is going into your Google Drive, okay? So you should